Welcome to The Deep Dive, your shortcut to understanding the cutting-edge science that is really starting to change how we think about the human body. We've got quite a stack of research here today, and we are plunging into a world you actually can't even see, the world of exosomes.
Yeah, exactly. And these aren't just some obscure biological bits and pieces. They are tiny, truly nanoscale messengers produced by pretty much every cell in your body. If you've heard of things like hormones or neurotransmitters, think much, much smaller, but maybe even more complex. We're really talking about the body's essential global communication network, kind of like a biological internet, you could say.
A biological internet, I like that. So our mission today is to really pull out the fundamental science behind these cellular messengers. You know, what are they? How do they coordinate function across basically every organ? We'll look at their, frankly, dramatic potential for regenerative medicine. And maybe the most surprising part, their direct role in linking things like psychological states, stress, even mindfulness, directly to physical health and aging. It feels like the mind-body connection finally getting mapped out at the molecular level.
It really is a paradigm shift in cell biology, absolutely. If you've ever wondered, how your thoughts might physically show up in your cells or how expectation can become a physical reality, well, this looks like the molecular pathway.
Okay, right. Let's unpack this then, starting with the absolute basics.
Yeah.
What exactly are we dealing with here? What is an exosome, like structurally?
Okay, so at its simplest, an exosome is a type of lipid vesicle. Think of it as a tiny sac bound by a membrane secreted by cells all over the body, like a microscopic bubble, sort of self-propelled, and it's filled with very specific information. They're actually part of a bigger family called extracellular vesicles, but exosomes are, well, the smallest and you could argue the most sophisticated of the bunch.
And how microscopic are we talking? Give us a feel for the scale.
Oh, they're incredibly small. The range is typically between 30 and 150 nanometers in diameter. So to put that in perspective for you, a typical human cell might be around, say, 10,000 nanometers wide. So an exosome is roughly 1,000th the size of that cell. They're so tiny you can't see them with a standard light microscope. You need things like electron microscopes or special tracking tech just to even, confirm they're there.
And what's really amazing is their history, isn't it? I mean, for decades, they were just completely misunderstood, pretty much ignored by researchers.
Right. That's one of the kind of great ironies in biology. For over 30 years, when scientists saw these little sacs being pushed out by cells in lab cultures, they often just dismissed them. They were thought of as just cellular debris or waste. Some researchers, apparently in their lab notes, even sort of jokingly called them junk or cell poop.
A very unglamorous beginning for the body's internet.
Exactly. But that view has been completely turned on its head, really starting around the mid-2000s when we began to figure out what was actually inside them. We now know they're the opposite of junk. They are these highly sophisticated, absolutely crucial packets of functional information.
Okay, so let's get into that complex cargo they carry. Because it's way more than just a simple chemical signal, right? Which is why they've kind of upended the old cell communication model.
The cargo is precisely what makes them so powerful. An exosome acts like a really precise nanoscale snapshot of its parent cell's current state, its metabolic state, its genetic activity. The contents are super diverse. You find growth factors, various proteins, often things like heat shock proteins, which help with stress survival, or enzymes, lipids, and really critically, different types of functional RNA, specifically microRNA or miRNA, and messenger RNA, mRNA.
So if we compare that to a classic signal, like a hormone, which is usually like one specific key for one specific lock, what's the big difference in function here?
Well, the difference is really in the complexity and the depth of the instruction. If a classic signal molecule is maybe a single word like grow or stop, an exosome is more like a whole paragraph. Sometimes it's even like a complex software update for the receiving cell. It delivers this multifaceted signal package. This allows it to influence a whole range of outcomes in the target cell, often simultaneously. Things like cell maturation, differentiation, how inflammation is regulated, even blood vessel growth.
So their main job is communication, but with really broad consequences across the whole system.
Yes, fundamentally, they're a method for cell-to-cell communication that allows the body to coordinate really complex processes. Take wound healing, for example. An exosome might carry instructions to promote new blood vessel growth, that's angiogenesis, while at the same time, it could be carrying instructions to tell nearby immune cells to calm down, to suppress excessive inflammation. So it's managing multiple messages at once.
That distinction, the fact that the message can be so multi-layered and reflect the parent cell's current status, that seems really crucial for understanding everything else we're going to talk about, especially aging and stress.
Hmm.
Okay, so if these tiny complex messengers are constantly being released by cells everywhere in the body, we must be looking at a truly massive integrated network. How does this messaging system actually work? What gives it that name, the systemic communism?
Yeah, the systemic communisome, it really refers to the collective action of all these exosomes establishing this dynamic body-wide information network. It flows in multiple directions. It's a coordination system that really integrates the nervous system, the endocrine system, the immune system, all on a scale that, honestly, we couldn't properly explain at a molecular level before. It's like a biological internet where information flows quickly and specifically, even between distant organs.
But for that information to flow effectively, the packets need delivery mechanisms, right? The research points to 3 distinct ways they deliver their message, which sounds way more sophisticated than just a simple key in a lock.
That's right. And the way an exosome actually interacts with the recipient cell really determines how big the effect is and how fast it happens. Cells don't just use one method. They seem to use these 3 pathways depending on, how urgent the message is and what kind of message it is.
Okay, let's start with the first one, the one that sounds a bit like classic signaling, direct surface interaction.
Right. So in this case, the exosome doesn't even need to go inside the target cell. Proteins on the outside of the exosome, they're called ligands, bind to specific receptors on the surface of the recipient cell. This is pretty similar to how a hormone might work. That binding triggers an immediate signal cascade inside the recipient cell, causing a very rapid response, maybe a change in metabolism, or it tells the cell to release another molecule right away. It's fast, it happens right at the surface, a quick instruction.
Okay, the second way, membrane fusion, sounds much more direct, more impactful. Is this maybe for more critical messages?
It seems like it's used when the parent cell needs to deliver functional tools, like right now. In this case, the exosome membrane actually fuses directly with the target cell's outer membrane, essentially merging into it. This dumps the entire contents of the exosome, all the proteins, the functional mRNA, the microRNAs, straight into the recipient cell's cytoplasm. It's a really efficient way to deliver molecules the recipient cell might need immediately, perhaps to repair itself or make a drastic change in function.
And then the third way, endocytosis, where the recipient cell actually pulls the messenger packet inside itself.
Correct. That's internalization, or endocytosis. The recipient cell actively engulfs the exosome, pulling it into an internal compartment, usually an endosome. Once it's released from that internal bubble, the exosome's cargo, particularly the RNA, can really get to work. The messenger RNA can be translated into new proteins the cell needs, or the microRNAs can go and regulate which genes are turned on or off. This pathway often leads to the most profound, sort of longer-term changes. It's essentially like reprogramming the cell's behavior over time, giving it new software to run on.
That really helps explain the sophistication. You've got speed with surface interaction, immediate tool delivery with fusion, and then this long-term reprogramming via endocytosis.
But how does the system handle distance? How do these messages travel far across the body and actually know where they're supposed to go? Right, the long-range signaling, that's mainly managed through the circulatory system. Exosomes travel through the blood and the lymph fluid, acting a bit like endocrine signals, like hormones, but potentially with much more precision. The sheer scope of communication between different organs that they seem to manage is pretty staggering.
Can you give us a solid example of this kind of endocrine-like action?
Sure. Think about the metabolic conversation between, say, your liver and your fat tissue. Exosomes that are released by the liver can carry specific microRNAs. These travel through the blood to your adipose tissue, your fat cells, and they can actually tell those cells how to store lipids, how much fat to store, and it goes the other way too. Exosomes released from fat tissue can travel to the pancreas or to your muscles and influence how sensitive those tissues are to insulin. It's this constant multidirectional chat coordinating how your body manages energy across different systems.
That makes total sense. Organs aren't just operating in isolation. But hang on, if every cell is constantly sending out these messages, how does an exosome from, say, a stressed liver cell know it needs to target a fat cell and not like a kidney cell? That sounds like it needs some pretty advanced targeting.
It absolutely does. And exosomes aren't just randomly flooding the system. Very often, they're equipped with specific proteins or lipids on their surface that act like molecular zip codes or address labels. These surface markers allow them to preferentially bind to and then deliver their cargo to only specific types of cells or specific organs. This helps ensure the message, whether it's store more fat or boost mitochondrial function, actually gets to the right destination. It makes the communication both system-wide and highly targeted.
Okay, this leads us perfectly into what might be the most dramatic finding. This dual role exosomes play in aging and rejuvenation. Section 3 really dives into how these packets are either, spreading the bad news of aging or delivering the good news of youth.
They absolutely are. They're kind of the ultimate molecular double agent in the aging process. They can promote aging, even speed it up, or they can offer really incredible regenerative effects. And the outcome depends entirely on where they came from, the state of their parent cell, and what specific cargo they're carrying inside.
Let's start with the darker side then. The pro-aging effects. How do exosomes actually spread senescence? We hear this term zombie cells thrown around a lot, but how do they actively recruit new zombie cells?
Right. So when cells get old or damaged beyond repair, they can enter this state called cellular senescence. They stop dividing permanently, which is good in one way, preventing cancer, but they don't die. They hang around and become metabolically active in a harmful way. These zombie cells start spewing out this toxic, highly inflammatory mix of molecules. It's known as the senescence-associated secretory phenotype, or SSP for short, and exosomes are now understood to be a really critical part of that SSP package.
So the zombie cells aren't just sitting there passively, they're actively broadcasting this like sickness signal.
Precisely. These pro-aging exosomes are loaded with pro-inflammatory cargo, various damaging proteins, and specific detrimental microRNAs. When these exosomes get taken up by nearby healthy cells, they can actually trigger what's called secondary or bystander senescence. They effectively sort of infect healthy tissue with the aging signal. This spreads the aging phenotype, causes chronic low-grade inflammation, which is a hallmark of aging, sometimes called inflammaging throughout the tissue, and eventually can impact the whole organism.
Wow, that's actually a terrifying thought that aging is almost communicable at the cellular level through these packets. But okay, thankfully there's the flip side, the anti-aging potential. Where do the beneficial exosomes typically come from, and what kind of instructions do they carry?
The beneficial ones usually come from young, healthy cells. Particularly potent sources seem to be mesenchymal stem cells, or MSCs. You can also get them from perinatal sources like amniotic fluid. These youthful exosomes carry cargo that's fundamentally restorative, things like specific regenerative proteins, maybe even the enzyme telomerase, which helps maintain chromosome ends, and a beneficial mix of mRNAs and miRNAs.
And what specific signals, what sort of molecular factory reset instructions do they give to old cells?
The instructions are pretty comprehensive, targeting the main ways cells break down with age. They basically tell aged cells to reduce oxidative stress, which is a major source of cellular damage. They instruct them to enhance mitochondrial function, get the cell's power plants working better, which counters that age-related energy decline. They also promote cell repair mechanisms and send anti-apoptotic signals, trying to prevent premature cell death. And crucially, as you mentioned, they can deliver factors that help stabilize telomeres. Those are the protective caps on our chromosomes that shorten with each cell division, contributing to aging and genomic instability.
Yeah, that detail on stabilizing telomeres seems key. It's not just patching things up, it's potentially reversing one of the fundamental molecular clocks of aging.
It is, and a lot of this research was really spurred on by those famous parabiosis experiments, where they surgically joined the circulatory systems of old and young mice and saw the old mouse get rejuvenated. Exosomes are now considered probably the key mediators, the main signaling molecules responsible for that effect.
The sources mentioned that actually injecting exosomes from young animals into old ones reversed some key gene expression patterns related to aging. What specific molecular proof did they find for this reprogramming?
They saw a really clear downregulation, a decrease in core senescence markers, things like the protein P16INK4A, which is like a classic flag for cellular aging. And at the same time, they observed an upregulation, an increase in genes related to telomerase activity and genes involved in DNA repair pathways. This is pretty strong molecular evidence that the cargo inside those young exosomes can actively push old cells back towards a healthier, more youthful state.
Now, okay, this is where I have to jump in with a bit of skepticism, which is always important for a deep dive like this. The sources mentioned some very recent quadramatic experiments showing significant lifespan extension using engineered exosomes, up to a 15.4% increase in median lifespan in aged mice. I mean, 15% increase is huge. How much of that is just mitigating severe disease versus genuinely extending healthy lifespan? And what are the limitations we need to consider with that kind of engineered delivery?
That's an absolutely essential question to ask. That 15.4% increase, while it sounds incredible, was achieved using exosomes specifically engineered in a lab to carry a very potent molecule, a microRNA called miR-302b. The main mechanism they identified was indeed a profound reversal of cellular senescence in several key organs. And yes, it did lead to measurable improvements in physical health markers in the mice, like better balance and grip strength, which does suggest healthier aging, not just keeping sick mice alive longer. But, and this is a but translating that precise engineered exosome, that specific dosage, and that delivery system for mice into the complexity of human biology is a massive leap. It confirms the potential of targeting senescence via exosomes, but it absolutely doesn't confirm immediate human application is around the corner.
So the principle seems sound targeting senescence works, but getting it to work safely and effectively in humans is still a huge challenge.
Exactly. It identifies a very promising target, the spread of senescence by exosomes, but the optimally engineered and delivered human version. That's still very much in the research and development phase.
Okay, and finally, in this section on aging, let's quickly touch on this idea of the transplantation tug of war. This sounds like where the whole exosome communication system is really put to the test in a real-world scenario.
Yeah, this hypothesis is fascinating. Imagine you take an aged organ, maybe a kidney, from an older donor and transplant it into a young, healthy recipient. What happens next is likely this dynamic two-way communication battle involving exosomes. The young recipient's healthy body starts sending out its youthful exosomes. They travel to the newly transplanted aged organ, trying to deliver regenerative instructions, trying to, fix it up or rejuvenate it.
But the aged organ itself is fighting back, right?
Yeah.
Because it's full of those senescent cells pumping out their inflammatory SAP signals.
That's the tug of war, precisely. The aged organ is releasing its own aged exosomes, packed with those pro-inflammatory SAC factors. These aged packages travel out from the organ into the young recipient's body, and they can potentially induce bystander senescence in the recipient's own healthy tissues, maybe their liver, muscles, immune cells, potentially speeding up the biological aging of the young recipient. The ultimate outcome, how long the organ lasts, the overall health of the recipient might really depend on which set of exosomal signals wins this molecular conversation.
It paints a really vivid picture. Exosomes are essentially the currency, the medium of exchange for biological age transfer between tissues.
All right, let's shift gears to medical applications. This is where, the huge promise meets some necessary caution. Exosomes seem to offer enormous potential for regeneration, especially in repairing damaged tissues. They really are powerful amplifiers of the body's own healing response. Clinically, they're showing great promise because they can boost healing after surgery or in reconstructive procedures. They do this mainly by influencing blood vessel growth and regeneration, that angiogenesis process we mentioned. Plus, they bring these potent anti-inflammatory effects that's critical for better wound repair, especially in difficult cases like chronic wounds, severe burns, or even managing inflammation after cosmetic procedures.
And the sources really highlight their use in joint problems, particularly osteoarthritis. And there was a key insight there about treating the cause, not just the symptom, right?
Yes, that's the really important takeaway there. Osteoarthritis is often thought of as just, cartilage wearing away from use. But a lot of current research suggests the cartilage breakdown is actually the result of underlying problems.
Yeah.
Specifically, metabolic and inflammatory changes in the chondrocytes. Those are the cells that make cartilage located right at the edge of the bone. These cells become sort of cranky, senescent, and highly inflammatory as part of the disease process.
So if you inject exosomes, you're not just trying to spackle over the worn cartilage surface. You're aiming to reprogram the cells that are causing the problem in the first place.
Exactly right. You're delivering that factory reset signal to those inflamed, dysfunctional chondrocytes at the bone edge. Studies are showing that injecting the exosome therapy into that bone edge area where these critical cells live seems to give much better, longer-lasting, truly regenerative results compared to just injecting into the joint space itself, which might only address the downstream symptoms like worn cartilage or pain. The goal is to reverse the underlying metabolic dysfunction that's driving the degeneration.
And their tiny size gives them a massive leg up over traditional drugs when you're trying to treat neurological disorders.
Absolutely. This is often called the holy grail in neurobiology, getting treatments effectively into the brain. Because exosomes are so incredibly small, down to 30 nanometers, they seem to have this natural ability to cross the blood-brain barrier, the BBB. The BBB is normally super restrictive. It's designed to keep almost everything out of our central nervous system for protection. So the fact that exosomes can get across makes them incredibly promising as potential delivery vehicles. You could load them up with specific neuroprotective drugs or RNAs and shuttle them directly into brain tissue to treat things like Alzheimer's, Parkinson's, maybe even brain injuries.
They're also making waves in dermatology and aesthetics, everything from calming inflammation to promoting youthfulness. But we need to be specific about how they're working here, not just list applications.
Right. In the cosmetic and dermatology world, the applications mostly rely on their signaling power. For skin rejuvenation, for example, they seem to enhance the proliferation of fibroblasts. Those are the key cells that make collagen and elastin, the stuff that keeps skin firm and elastic. They also seem to decrease the rate at which fibroblasts become senescent, or old and non-functional. So essentially, they're telling the skin's factory workers to ramp up production of good stuff and clear out the old, ineffective workers. This leads to increased collagen and improved skin quality.
I found the connection drawn between melasma and aging particularly interesting because it ties right back to that idea of fixing the underlying structure.
Yes, melasma, those patches of darker pigmentation, is often just seen as a pigment problem. But there's a growing view that it's actually linked to an aging process in the skin, specifically a weakening of the basement membrane, that crucial layer between the epidermis and the dermis. And there's often chronic inflammation involved, too. Traditional treatments might just target the pigment itself. Exosomes, though, offer the potential to actually help repair that weakened basement membrane structure and calm the underlying inflammation. It's a more fundamental approach aiming for longer-term improvement. They also show promise for hair loss, apparently by signaling hair follicles to shift into the growth phase and increasing growth factors around the follicle area.
Okay, now this is the really crucial part of this whole discussion, especially with all the products popping up. The cautionary landscape. All these amazing results are mostly preclinical, yet the cosmetic and anti-aging industries are already heavily marketing exosome products. We really need to hammer home the reality check here.
Yes, we absolutely must be extremely cautious. The vast majority of the really exciting results we've been talking about, the lifespan extension in mice, the potential disease reversal, they come from preclinical studies. That means cells in a dish or small animal models like mice. The critical issue, the gap, is that these results often fail to translate effectively into reliable, safe human treatments. There are significant manufacturing, delivery, and consistency challenges.
So what are the key knowledge gaps that someone considering these treatments needs to be really aware of right now?
Well, there are major scientific and regulatory hurdles still. First, the source material is a big issue. Are the exosomes in a product derived from, say, synthetic sources, or perinatal amniotic fluid, or mesenchymal stem cells? The exact cargo, that specific mix of microRNAs and proteins, can vary wildly depending on the source cells and how they were cultured and processed. A product from one company might be totally different from another. Second, stability and storage are tricky. Exosomes are basically fragile little bubbles of fat. They need strict cold chain storage to stop them from degrading. Many commercial products might not meet those standards, meaning what you get might not even be active.
And the delivery method itself is often completely ineffective in many of the consumer products being sold, right?
Yeah. That is perhaps one of the biggest areas of, well, let's just say misleading marketing right now. Simply rubbing a serum containing exosomes onto your skin or sprinkling it on is often pretty ineffective. Why? Because exosomes, while tiny, are generally too large to just passively seep through the stratum corneum, which is the tough, outermost protective layer of your skin. To get them where they need to go to have a biological effect, they usually need some kind of mechanical help, things like microneedling, maybe certain laser treatments, or direct injection. These methods create temporary channels through the skin barrier, so often a good chunk of any benefit seen might actually be from the controlled injury caused by the delivery method itself, which triggers natural healing rather than the exosomes alone.
So using that grocery bag analogy from the outline, buying a generic exosome serum and rubbing it on your face is kind of like getting a bag full of gourmet ingredients delivered, but then just leaving it on your doorstep instead of bringing it inside and cooking with it.
It's a pretty good analogy, yeah. Just having the ingredients, the potentially powerful exosomal cargo, isn't enough. They have to be delivered in the right concentration, probably at the right time relative to other factors, maybe alongside necessary cofactors, to actually get the desired effect. And honestly, We still lack solid clinical data in humans about what the optimal dosage even is for most conditions.
And we also really need to address the risk of unintended consequences. We talked about how exosomes can promote regeneration, but that kind of growth signal isn't always good, is it?
This is perhaps the most serious potential downside, especially for unregulated commercial products. Exosomes are powerful signaling packets. They can tell cells to divide, grow, or suppress inflammation. Now imagine someone has, say, an undiagnosed precancerous lesion on their skin. If they apply a potent regenerative exosome product to that area, is there a risk that those growth signals could actually accelerate the proliferation of those abnormal cells? Could it potentially promote skin cancer development? We simply do not have enough rigorous human safety data yet to rule out these kinds of negative outcomes. Until the source, the cargo, the targeting, and the dosage are precisely characterized and controlled through proper clinical trials and regulation, extreme caution is really warranted. The underlying science is incredible, no doubt, but the widespread application is still very much an open question that demands much more high-quality human research.
That reality check is so important. Okay, now let's move into what might be the most profound section of this entire deep dive, the exosome psychology axis. This sounds like the actual molecular explanation for the mind-body connection operating within that field of psychoneuroimmunology, or P&I.
Yeah, this section really does fundamentally change how we should think about health and disease. Exosomes are increasingly recognized as this critical molecular link connecting our psychological states directly to our physical health. Their ability to cross the blood-brain barrier is key here. It allows them to facilitate communication back and forth between the nervous system, the endocrine system, hormones, and the immune system. They relay signals in both directions. Top-down, meaning from the mind or brain down to the body, and also bottom-up, from the body back up to the brain.
Okay, let's start with that top-down signaling. How does psychological stress physically change the actual cargo inside exosomes? It's almost like stress triggers this very specific systemic molecular broadcast of, well, stress.
It really does seem to work that way. When you experience acute psychological stress, think public speaking, a sudden shock running right alongside that rapid spike in stress hormones like cortisol, there are profound changes happening at the cellular level. This leads to a measurable increase in the sheer number of exosomes circulating in your blood, and also a complete shift in their cargo profile, what they're carrying inside.
And the research even suggests that the presence of exosomes might be functionally necessary for the stress response to work optimally, like they act as some kind of molecular cofactor.
That's a really key finding from some studies, yes. Researchers found that if they experimentally inhibited the body's ability to produce exosomes, it significantly reduced not only the levels of circulating exosomes, but also the levels of stress hormones, the glucocorticoids. This strongly implies that exosomes act as an essential intermediate signal. They somehow facilitate the optimal release of cortisol from the adrenal glands. They seem to be a crucial molecular bridge connecting the immediate stress perception in the brain and cells to the body's systemic endocrine stress response.
Okay, so if stress is the input signal, how does this molecular broadcast physically translate that subjective feeling of stress into objective, measurable inflammation throughout the body?
It happens largely through this concept called sterile inflammation. Basically, cells under stress start releasing exosomes that are loaded up with specific molecules called damage-associated molecular patterns, or DAMPs. They also carry things like heat shock proteins. DAMPs are essentially the body's internal alarm signals. They're usually released when cells are damaged or dying, and they scream danger to the immune system.
So the DAMPs are broadcasting, "Houston, we have a problem," even if there's no actual infection.
Exactly. And at the same time, psychological stress tends to reduce the amount of certain immunoinhibitory microRNAs that get packaged into those exosomes. These are miRNAs that normally help to dampen down immune responses. So the exosome effectively sends out two messages simultaneously. We're under attack, signaled by the DAMPs, and don't hold back the defenses, signaled by the reduction in those inhibitory miRNAs.
And this combined signal unleashes inflammation systemically, even without any actual pathogen present.
Yes. And this provides a direct molecular mechanism for how chronic psychological distress gets converted into objective, chronic, low-grade systemic immune activation, that sterile inflammation we talked about. This is likely a major way chronic stress contributes to accelerated aging and a whole host of inflammation-related diseases.
Wow. Okay, that clearly explains the top-down effect. Now let's flip it for the bottom-up signaling. How signals originating in the body, packaged in exosomes, can travel up and influence the brain, our mood, and emotions.
This is absolutely fundamental to understanding why emotions often feel so physical, why we talk about gut feelings. Signals from the body definitely influence the brain's emotional and neurological state. For instance, if your body is experiencing inflammation somewhere, maybe from an injury, an infection, or a chronic condition like arthritis, the immune cells involved release pro-inflammatory exosomes. Because these can cross the blood-brain barrier, they travel up to the brain. There, they can contribute to what are known as sickness behaviors, that feeling of fatigue, low motivation, social withdrawal, and even the development of anxious or depressive moods that often accompany physical illness.
And exosomes are now being directly implicated in major mental health conditions like major depressive disorder, MDD. Can we actually trace this down to a specific molecular pathway involving immune cells within the brain itself?
We're starting to, yes. Research using models of MDD has observed increases in specific types of exosomal cargo, like a particular microRNA called miR95P, originating from peripheral cells. This specific cargo, when it reaches the brain, seems to push the brain's own resident immune cells, these cells called microglia, towards what's known as the M1 phenotype.
And the M1 phenotype is the aggressive, highly inflammatory, potentially damaging state, as opposed to the M2 state, which is more focused on anti-inflammation and repair, right?
Exactly right. M1 microglia activation leads to the release of neurotoxic factors, contributing directly to neuroinflammation, potential damage to neurons, and the pathology underlying depression. The exosome provides the delivery mechanism, the way that signals from peripheral inflammation or stress get delivered right to the brain's immune system, potentially turning it against the brain itself.
This all seems to culminate in making exosomes the strongest candidate we have so far for the actual molecular mechanism behind the placebo and nocebo effects, which is really the ultimate test of the mind's power over the body's physical state.
It provides a plausible way to connect psychological expectation directly to physical reality at the cellular level. If a patient truly believes a treatment will work, the placebo effect, the brain might trigger the systemic release of healing exosomes. These could deliver anti-inflammatory signals or perhaps analgesic, pain-reducing signals directly to the affected area, generating a real, measurable physical improvement. Conversely, if a patient strongly expects negative side effects, the nocebo effect, that negative expectation might trigger the release of negative exosomes carrying pro-inflammatory or other detrimental signals, literally helping to manifest the anticipated adverse physical effect. The exosome becomes the physical messenger carrying the instructions dictated by psychological expectation.
Which means our mental state isn't just some subjective feeling. It's literally dictating the content of the molecular communication packets our cells are constantly sending out. So if chronic stress makes those messages negative, can positive behavioral practices actively make the messages positive?
Absolutely. And the research looking at practices like mindfulness-based stress reduction, or MBSR, is incredibly compelling on this front. It's showing that conscious behavioral and psychological practices can indeed positively alter the cargo found inside exosomes. This demonstrates that our conscious efforts can induce sustained, beneficial molecular changes.
What specific changes have they actually observed in exosome cargo after people practice mindfulness?
Well, studies have linked MBSR practice with increased levels of a neuroprotective microRNA called miR29C found in exosomes derived from neurons. This specific miRNA has been associated with things like improved synaptic plasticity, how well brain connections adapt, and better cognitive performance. But perhaps the most profound finding relates to epigenetics, the control of gene expression. Mindfulness practice was shown to actually modulate the levels of DNA methyltransferases, or DNMTs, found within neuronal exosomes.
And DNMTs are those key enzymes that regulate epigenetics.
Yeah.
They control the long-term programming of which genes get turned on or off. That's fundamental.
Exactly. The fact that a psychological intervention like mindfulness can change the exosome-mediated transfer of these core epigenetic regulators is really significant. It suggests that positive psychological input gets converted into functional molecular instructions. These instructions can then potentially induce sustained positive changes in gene expression and the long-term function of the recipient cells, particularly within the central nervous system. So mindfulness, from this perspective, isn't just about feeling calmer. It appears to be a genuine molecular intervention that changes the literal instructions your brain cells send out to each other, promoting long-term brain health.
This entire deep dive really brings us back to one central powerful takeaway, doesn't it? Exosomes are like the cell's daily data packets. They're constantly reporting on status of every cell, creating this global messaging system that influences aging, drives regeneration, and critically provides a tangible mechanism for how our thoughts and feelings translate into physical reality.
The clinical potential of understanding and eventually manipulating this exosomal communication access is just enormous. We're looking at the possibility of non-invasive biomarkers, maybe testing blood or saliva for specific exosome signatures to track biological age or monitor disease progression, especially in hard-to-access places like the brain. And of course, there's the huge potential for developing new therapeutic strategies, maybe using engineered exosomes for highly targeted drug delivery, leveraging their natural ability to travel almost anywhere in the body and cross barriers like the BBB.
And as this research continues to really validate the power of the mind-body connection at this incredibly detailed molecular level, it kind of forces us to rethink causality, doesn't it? I mean, if the exosome is the physical mechanism linking mind and body, and its cargo is directly influenced by our past mental state, our chronic stress, our positive beliefs, our mindfulness practice, what deeper connection might these vesicles have to some ancient concepts? Ideas like a vital life force or whole body energy signaling, maybe concepts like qi in traditional Chinese medicine.
It definitely raises some fascinating, almost philosophical questions. If the placebo effect genuinely works, at least in part by generating beneficial exosomal instructions based on belief, is the exosome the first piece of concrete molecular evidence suggesting that ancient systems describing things like qi were actually observing a real nanoscale physical communication network mediated by our internal state, including expectation and intention? It makes you wonder, doesn't it? How much of your current physical reality right now is simply the accumulated result of all the exosomal messages sent out by your past mental and emotional states?